Hello, and welcome to the Find Hosted Study Files in the Data Browser video module, which is a part of the 7 Bridges onboarding lesson. In this video, you will learn all about the data browser and how it can help you connect your projects to the datasets that are important to you. While watching this video, please keep in mind 2 key concepts. The CGC provides access to over 3 petabytes of publicly available data as part of the Cancer Research Data Commons, or CRDC, ecosystem. The CGC provides access to both controlled and open access data, which equates to thousands of files spanning dozens of disease types. At the end of the video, you will be able to find and filter hosted study data files, link hosted files to your projects, access open data for testing and result validation, and lastly, understand which datasets are open and which are controlled. For the demonstration today, we will be doing 2 things. 1, we will query OpenTCGA datasets for genomic files, and 2, we will query the OpenTCGA dataset for tissue type-based multi-sample genomic files. Okay, I've switched tabs, and as you can see, I'm already logged onto the CGC powered by 7 Bridges. I'm in a project I made for this demonstration called Onboarding Tutorial, Data Browser. The goal is to demonstrate Data Browser and how to access files which are a part of open datasets. So our first goal, as I wrote down in the description, is to find genome data and add it to this open data project. One clue that this is an open data project is that there's no lock icon with the word controlled next to the name of the project. So let's go ahead and get started. To get access to the data browser, go to the drop-down menu at the top and find where it says Data. From here, click on it and select Data Browser. Now the great thing about Data Browser is you can develop some pretty complex queries where you're selecting more than one option and finding the maximum number of specific files. For this demonstration though, I'm just going to pick one project and then click Explore One Selected. Now, if I had some saved queries, that would appear here. But since we're starting from scratch, we are going to select the type of data and we're going to build off of that. You can, if you're interested, click on examples to view some already saved ones. Alright, I normally like to start with file when I'm working with this type of data, but you could start with any number of subjects, certain sample IDs, or even if you have a certain sample study you would like to look at. So I'm going to pick the file entry, and then from here, each of the rectangles represents an entity. So you know all the subjects would be an entity, samples are an example of an entity, and in this case, I have all the files here. From here, we're going to filter for certain properties. So the highest level is file, and then I'm going to scroll down and look for experimental strategy. And from here, we have numerous options. We have attack-seq, genotyping, methylation, whole genome sequencing. You can select whichever one you're interested in. For demonstration purpose, I'm going to select RNA-seq and filter by that. Now, for my next selection, I'm going to scroll down to data type. And here we have, again, many options. Aligned reads, we have gene expression quantification. If you hover your mouse over the text, a little box will pop up allowing you to read the full thing. And we have other options like transcript fusions, splice junctions. For, again, this demonstration, I'm going to click on gene expression quantification and click filter by that. So you'll notice now in the bottom left-hand corner that we have a number, 517,142 files, but this number is crossed out. What that's telling me is I need to refresh to get an updated count of files. So it went down now to 11,123. If you scroll down, you'll have a chance to look at some of these files. The green open lock symbol here signifies that this data is open data. If you're not sure, you can also click Access Level under File and filter only by Open Data. We could still look for data that is closed. However, when we go to copy that to our project, the system would programmatically check for dbGaP credentials and make sure that we have appropriate credentials. And if we did not have the appropriate credentials, since our project is an open project, it would block us from accessing the data at that point. For any of these files, you could select the file and learn more about it. You can also pull this up. So here we have information about access level, the date that it was created, data category, the format in which it comes. We have the experimental strategy, the size of the file, and some other information such as the genome build and the last time it was updated, for instance. So this information is called metadata, and it comes with all of our files. You can click on any of these to get different metadata. So if you know you're looking for a particular file, you can go ahead and click on it in the middle section, and it gives you the name of the file, again, as well as all the metadata that's associated. So I picked these files, which are gene expression quantification, and they come from RNA-seq. Once we're ready, we can select Copy Files to the Project, which is on the top right-hand corner of the screen. From here, we'll have to select the project we'd like to copy it into. I'm going to select our latest project, Onboarding Tutorial, Data Browser. However, if you're not seeing the full project list, you can always search and it will come up. So once I've selected this, I have the option to add tags. So I'm going to add a demo tag, maybe an RNA-seq tag, just for my own knowledge. And then once I'm ready, I'll copy selected files to the project. And a little pop-up appears here, copying files to this project, Tutorial Data Browser. So keep in mind, we didn't actually copy the data out of these CGC hosted buckets, but rather it created a symbolic link in our Files tab of the project that we specified. So let's go back to that project. So we'll click on the Projects tab, and then the top link here is Onboarding Tutorial Data Browser. From here, we'll click on the Files tab within our project. And as you can see, all the files were copied over to this project. You can select any of these files again to view information about the file, as well as to view metadata. So all of this information follows along with the file, regardless of what analysis you do with the file. It comes with case and demographic data, diagnostic data, and some other helpful information. E-tissue sample genomic data. So again, we're going to scroll up to where it says Data and select Data Browser. From here, you have the option to explore any of these datasets. I'm going to stick to TCGA for now and select Explore Dataset, or Explore when selected. And as you recall, last time we started with File, but this time I think I'm going to start with the case entity, just to show you guys what that looks like. So for this particular demonstration, I'm starting with case, and as you can see, we can filter by numerous options here. We have case identifier, the date, time that it was created, disease type, primary site, and some other options. I'm going to actually click on case and select that, and from here, I'm going to select sample. So now we have a whole new set of options. I'm going to scroll down and find sample type. And from here, I'm going to select primary tumor. Here we go. So now we have a set of samples coming from primary tumor. I'm going to go back and click on case once again and select another set of samples. So you'll notice the first sample was just called sample, and this one is sample 1. So sample 1, again, I'm going to scroll down, identify sample type, and from here I'm going to select solid tissue normal. And you can always click and drag these around if you can't quite see every option. So solid tissue normal is selected, and I'm going to filter by that. So now we have two sets of samples. We have primary tumor as well as solid tissue normal. I'm going to go back to this first sample box and select this to further refine. So I'm going to, from here, click on File. And then from File, I'm going to look for a data type, and I'm going to select gene expression quantification and filter by that. I'm also going to filter by experimental strategy and select RNA-seq. Now I'm going to do this with sample 1 as well. So for this box, again, I'm going to click on it and then select File. I will scroll down for data type, and I will select gene expression quantification, which is right here. Again, pull up if you can't see all of the options. And then once again, I'm going to filter by experimental strategy and select RNA-seq. Okay, so now we have cases, and these cases are split into two options here. So we have paired samples coming from primary tumor or solid tissue normal. And then these samples are further refined, and we're only looking for gene expression quantification coming from RNA-seq experiments. So we can scroll down, and again, as you can see, these numbers are crossed out as we've refined them further. So we can refresh to get updated numbers. And we have 726 files belonging to solid tissue normal and 701 files coming from the primary tumor. We can pull this up to find further information about these files. Again, you can select any of these files to read some of the metadata. And then once we're ready, we're going to once again go to the top right-hand side of the screen and select Copy Files to Project. I'm going to move them once again to my onboarding tutorial data browser project. From here, I'm going to use the same tags I set up before, but maybe I could add some additional ones so I can easily identify these files. Like maybe I can write tissue specific or something along those lines so I can easily search for them. Once I'm ready, I can copy the selected files, and again a pop-up happens. It says 701 of 701 files have been copied to the onboarding tutorial data browser. So once I'm ready, I can click on my projects, go back to that main project, and we can look for these files in the Files tab. And as you can see, we already have a full populated project with many files. A lot of these are from the earlier portion of the demo. So I'm going to search for the tag we used. So click on Tags and select Tissue Specific. And from here, we see only the latest files are listed. Again, we can click on any of these files to get some information, and we can view the metadata of this file. So we see that this particular file comes from TCGA. It belongs to a stomach as the primary site. And we have some other information, like this is a solid tissue normal sample. So some key takeaways from this demonstration is Number 1, you can find all study files for CGC hosted datasets in the data browser. 2, the green open lock icon indicates that the file is open to all users, so anyone can copy that into their project. 3, the red lock icon indicates that the file requires dbGaP approvals for the particular study the file belongs in, so you need to have controlled access and dbGaP authorization to copy those files. And number 4, controlled files can only be copied to controlled projects. This concludes the video. The next suggested video is called Bring Your Own Data, and we'll go into detail about how to upload or import your own data into your CGC workspace.